Good afternoon. I'm Stephen Crystal, Special Populations Core Lead, and I want to thank everybody for joining our final Special Populations Seminar of 2021. Keep your eyes open for future announcements of our Spring 22 series in the coming weeks. Georgette has done an amazing job this year in organizing such interesting and fruitful webinars. It's my pleasure to introduce our presenters, who are two distinguished psychiatrists, Carlos and Michele Pato, who recently come to Rutgers to oversee behavioral health, addictions, and genomic psychiatry research, a rapidly growing field in psychiatric medicine that investigates the genetic connections to mental health. Michele Pato is the inaugural director of the new Rutgers Center for Genomics of Psychiatric Health and Addiction, the RCPHA. Do you have an abbreviation for that, Michele?
Not yet. We're going to have to work on that.
Carlos is the first executive chair of psychiatry for RBHS. They each are professors of psychiatry at Robert Wood Johnson Medical School and Rutgers, New Jersey Medical School. Michele and Carlos, please take it away.
Okay, so Carlos is going to roll off screen for a minute and I'm going to get us started. I guess I have to start at the beginning, which is to say we've been doing this work for 25 or 30 years, been married for 41, so we're going strong. To date, we've collected a sample of 65,000 participants. And a participant can be anyone. It can be someone with the mental illnesses that we're most focused on, both schizophrenia and bipolar disorder. And it can be a control. And a control, that doesn't mean they don't have something. And in fact, we finally got our first obsessive compulsive disorder genomics grant, even though I've been studying OCD since I was at the NIH in 1986. But getting genetic study of OCD took some work. And some of my OCD subjects in that genetics work also have schizophrenia, because we knew in writing OCD grants that there were so many comorbidities that we couldn't exclusively say they had to have only OCD. But with schizophrenia and bipolar, it does have to be your primary diagnosis. So that tells you a little bit of how we got up to 65,000. In about 20, 25 years. However, even from early beginnings, we wanted anyone who wanted to participate to be able to participate. So back in 2006, when we were getting some substantial grant support while at the University of Southern California, we submitted a grant to collect 10,000 people with schizophrenia and 10,000 controls. And our collaborators, which were at 12 sites around the country, started saying to us, gee, we want to take anyone who comes in the door, and some of our sample is going to be of African ancestry, and some is going to be of Latino ancestry. Can we take those people too? So we wrote a little email to the NIH, and we said, listen, I know that most of the collection to date, which I'll talk about a little bit more in a moment, has been in European Caucasian ancestry, but why should we turn anyone away? And they said, please collect anyone who comes in. And that began our collection of diverse populations. We were so successful in collecting diverse populations with that first grant in 2006 that right before we got recruited to downstate, where we came in 2015, we got our first African ancestry recruitment grant. We'll talk a little bit more about that in a minute. Now, because of the large sample we've had, we've been able to do, she was on 50,000 so far, 20,000 of whom are of African ancestry. And we've also done whole genome sequencing on about 8,000. How do we do this? How do we make it happen? Well, one of the critical features to getting that grant in 2006 to do 10,000 cases and 10,000 controls was even though we, like everyone else, had dutifully been using the DIGS, the Diagnostic Interview for Genetic Studies that was developed by the NIMH in 1994, it took 3 or 4 hours to ask all those questions. And we began to question ourselves, did we need every single question answered? So we developed an abbreviated version of the DIGS called the DIPAD, the Diagnostic Interview for Psychotic and Affective Disorders, that included 83 of the questions from the DIGS. And then we had cohort of people who got both instruments, and we got the same diagnosis with good reliability and validity. So when they proposed us collecting 10,000 people with schizophrenia, we said, we can do it, but we can't hold people for 3 or 4 hours. We're going to use the DIPAD, and that way we can get the interview done within an hour or two. And they said yes. So time was an important element. The other thing that was really critical that we had already started back with our initial studies that we first got in 1994-96 in the Azore Islands of Portugal was we had a screening tool that assessed very briefly for everything else. And so we've now written a number of papers on probable psychiatric illnesses like PTSD, like panic disorder, like even OCD, and we have specific questions about substance abuse that really follow the CAGE questions as well. So we have 6 questions about alcohol use, 4 questions about tobacco use, 2 questions about cannabis use, and 2 questions about illicit drugs. Even though this instrument was developed in 1996, and we've had to tweak it a little bit along the way, we knew to separate cannabis and other illicit drugs, because as we now see what happened in the early 2000s is there's a question about is cannabis illicit or not, but at least it'll lead to more research. This is what we faced when we did our first research in 2006, was that even in 2009, 96% of the sample was of European Caucasian ancestry. So of course, pushing forward with our 206 study already collecting, I know, but in our own work, we knew that was happening. So we were glad that we had decided to also collect African ancestry, Latino ancestry. In 2016, it still was not very much African or Latino ancestry. The basic mix was now also 20% Asian. But thank God we got that grant in 2015, which we carried with us to downstate to begin to collect African ancestry. Now, as we've tried to analyze it, there are a couple of reasons for these problems. But let me tell you what we've managed to do now. Since 2006 and since 2015. So we got the first African ancestry and we dutifully collected for 6 years while we were at downstate. And then in 2020, while we're all sitting because of COVID, we kept busy by writing more grants, and we were issued 2 more grants, actually 3 in 2020. One was expanding the African ancestry collection, and the second was to begin Latino ancestry collection. So we came to Rutgers in early 2021 with both these grants ready to go. Some of these include samples of schizophrenia, bipolar, and screen controls, but that doesn't mean they don't have illnesses. And we also have developed, while we were at downstate, a collaboration with the VA Million Vet Program. So we'll have a way of accessing both African ancestry and Latino ancestry participants in the VA collections of 1 million vets. Carlos and I have also worn hats at the VA for about 5 or 10 years each in our various positions that we've researched through. So we know that the VA database is phenomenal and has been in existence. Our electronic health records, the VA has been doing it since the 90s. So, you know, they're coming up on 30 years. So the record has quite a bit of data in it that could be useful. We also come to Rutgers with 2 more new grants. One is the PUMAS Collaborative Study, which looks at African and Latino ancestry throughout the world. And we're contributing to that as well. And I also now have another OCD genetics grant that I'm doing that is looking at in South America at the whole Latino collection. Just the acronym that they're using is kind of cute. So let me tell you what it says. It says Latin American trans ancestry initiative of OCD, and so they actually call it Latino as their study, and it's all over South America, as well as South American descendants in America, especially in areas like Texas, where one of the major PIs sits, and that's Eric Storch. So this is where in the world we've collected samples. So we've been around. We've been to a lot of places, and with our longtime collaborator, Jim Knowles, we now have a huge Pakistani collection of both schizophrenia, bipolar, and control subjects in Pakistan. So in some senses, a homogeneous sample, as everyone is Pakistani, probably more homogeneous than what we've been collecting in America. This highlights some of the other places that we're now doing collection with the PUMA study and with the Latino OCD study. Now, it's not always easy to collect diverse samples. A number of the things that interfere with this collection are people even coming in for treatment, whether their sites, particularly in certain areas of America, but also in South America and Africa, have adequate care so that people come in for treatment. There's also the cultural issues of what is causing your psychosis. And depending on the beliefs that might be held by people for religious reasons or other reasons, they may not even see it as an illness. In our own beginning work, which we did on the Azore Islands off the coast of Portugal, I call it the Hawaii of the Pacific, of the Atlantic rather than Pacific. We had to build trust with the community. Prior to our arrival there had been some work with Machado-Joseph's disease, where a group of collaborators knew that it's very familial and had zoomed in to San Miguel, grabbed samples and left and never talked to people again. You can't do that kind of work, particularly in genomics, particularly in inheritance, and not have a way of collaborating with people and letting them know. So we knew of the distrust that existed when we started this work back in the 1990s on the Azore Islands. And what I think has made us so successful at all the sites we've had around the country and around the world is to communicate, give information back. We would have town meetings, we would have presentations, we would send copies of our papers, and we would have our collaborators there encourage that they share this data with their clinicians, etc. Even though we couldn't make it sample specific and tell each subject what the results were, we did want to communicate the data and begin to work in that way. There are sometimes challenges, particularly in communities where there might be poor clinical care with getting data out of records and things like that. And that was why the efficiency of our DIPAD and our screening tool at least gave us some basic features that we have on every single one of our 65,000 subjects. These are some of the cues, as I was saying, that we've done to try to draw people in and make them feel comfortable with participation. It also becomes very important to make sure we study these things. Some people will say, why do you need to study me? You got all those Caucasian people, leave us alone. And what we like to point out is that we can't really provide appropriate treatment if we don't understand what you share and what you don't share with other races. And here's an example of that kind of thinking, where if there happens to be a variant that is shared maybe in 2% of Caucasians, 5% of Latinos, but 80% of those with African ancestry, that particular variant might make a real difference, not only in responding to a specific treatment, or in the side effects, sometimes very costly side effects that you might get from taking that drug. But if we don't study people as cultures and races, we won't know what might be important to know about what their genome is contributing to their response to illness. And there are some specific examples that have come out, for instance, in the antipsychotics and first and second generation antipsychotics that show in those of African ancestry, some of the second generation antipsychotics are quite toxic and don't work, whereas the first generation antipsychotics might work better. So Why did we come to Rutgers? We like contributing to communities. We like making a difference, and we like going and spreading our impact in other ways. And Rutgers is in New Jersey, and it's a huge population, but Rutgers itself as an institution spreads over most of it. But there's also opportunity that we haven't cultivated yet at Rutgers, now that we're on the Rutgers team, as I like to say. There are over 1 million people in New Jersey who probably suffer from either behavioral health or addiction problems. Yet those in acute treatment in the Rutgers system is only about 80,000. And our call centers are getting 50,000, 500,000 calls a year, at least in the last year. And so there is a gap here that we think could be very important to meet, both by engaging people in research and, hey, engaging them in treatment. We have the experience in our many recruitments when we were in downstate, where some of our interviewers actually, in talking with patients, particularly those with schizophrenia and bipolar illness, would ask them all these questions, and then they would say to them, well, gee, you know, how's it going? Or you would, oh, I'm not getting any treatment right now. Those doctors, they can't do anything. But then as they talked and they saw the kind of questions we were asking, we were able to help them engage. And we got thank you notes about deciding maybe they should get back to their doctor or engage for the first time in treatment, and not seeing it as a stigma, but as something they could do something about. So with that background, we came here to Rutgers, and I've taken over. This is now the third new center that I've started. I did one at USC for 10 years, which was a center for genomic psychiatry. And then I went to downstate and I ran an institute for genomic health. And why did I switch from genomic psychiatry to genomic health? You know, genetics doesn't just make you sick, it keeps you healthy. And so in coming here, I wanted to make sure we continue to emphasize that studying your genes isn't just finding out what's going wrong, it's finding out what's going right too. So we've started a center here on the genomics of psychiatric health and addictions. And I've set up 4 programs that are beginning to explore this issue. And in setting up these programs, I've hired some great team players. The first is a new director who we recruited from VCU, who's running the genes, environment, and neurodevelopment in addictions. She's had a lot of experience in the COGA programs while she was at VCU. Her name is Jessica Salvatore. And we're continuing to collaborate with one of our close collaborators at Downstate, Jackie Myers, who happens to know Jessica because they were at VCU together back about 10 years ago. And we're beginning to do some of Jessica's interesting work is looking at couples and the impact of alcohol use by one of the members of the couple on the other member and how that affects family and development. And she also has a genomic spin to her work. So then looking at what passes on to the children and what was in the grandparents becomes very important. The second program that we've already begun is with a wonderful person we recruited, Jennifer Michele from Emory. And some of you may know her husband, Michael Zwick, because he's the new head of
Senior Vice President for Research.
Senior Vice President for Research for all of Rutgers. So we have both of them here. And Jen is has cut her teeth on being the expert in 3q29 deletion, which is what looks like an advanced form of autism with pretty good interactions and stuff in children. But 70% of those children with the deletion develop schizophrenia in their 20s. And so her scope is broad in terms of understanding the phenotype and then understanding what happens in developing illness and looking at it from a genomic and molecular point of view. We have 2 other programs that we're cultivating. One is to look at multi-ancestry psychosis. And the research we've done in that has been with our collaborator who's still at Downstate, Tim Bigdley. And Carlos is going to give you a few more details about what some of that work has already shown and why we're so interested in pursuing it here. And we are in the process of recruiting someone else to Rutgers as well, who might help as a director in engaging this as we continue collaborations with Tim. And finally, it's what we call the POP GEM program, and this is population genetics and environment and how it affects mood. We've stayed more in schizophrenia and bipolar, both for joining with psychosis and mania. And we've done a little bit, as I said, also with OCD, but we hadn't really stepped into the depression arena. Given the heterogeneity of that sample, we were waiting for a time to expand into it. And now with coming to Rutgers, we're hoping to start a program in that area. And at the present time, we have done some work with Roseanne Peterson, who is at VCU as an adjunct professor, in understanding the impact here. And again, remember, genes and environment are both important. One of the things I always like to say when I talk with potential participants, and they say, oh, I don't want to know what my genes are. I don't want to know where I'm going to end up. I always like to say, you know, genes is not about destiny. It's just about risk. And the better we understand your risk, the more we can do to keep you healthy. And we all know, those of us who study genetics, that even in monozygotic identical twins, they're not totally concordant for an illness. One may get it and the other doesn't. So environment makes a difference. I'm going to turn the baton over to Carlos now, and he's going to talk a little bit more about the science. But before we go ahead, any burning questions at the moment, or do you want to hold them to the end? Not hearing anything, not seeing any hands, nothing? Okay, I'm going to turn over to Carlos.
Yeah, I think it's good to go forward. I have a number of questions, but I think maybe we'll save them for after Carlos speaks.
Okay.
Okay. Well, so I'm going to give you some brief summary of current results from these studies and then some of the way things have developed. So this is what we call a classic Manhattan plot. You see the skyscrapers. And what it is large-scale whole genome association studies, what we call GWAS, lead to, as the populations grow, increasing numbers of associated and verifiably associated common variants. And from these, we generate something we call a polygenic risk score, which can help define the higher risk population versus the lower. I will throw out a concept that we don't address in this presentation, but we also believe that there's a different type of PRS, which is a polygenic resilience score, so that we will be able to look not just at those things that increase your risk, but those things that are protective, and that's going to be an important element to the work we're doing. So this is a famous slide from 2008 when, 2014, when with 108 significant variants, we were able to demonstrate this output in schizophrenia. Significance in whole genome studies is when the association is at a p less than 10 to the -8 because of the obviously multiple testing that's occurring across the genome. What you see is as time has progressed, the numbers of associations have increased. In schizophrenia, we're approaching 300. In bipolar disorder, where the ends are smaller, we're in the 60s. And this is not just simply time, this is also an increase in the addressable population with adequate data to be able to analyze. Our own work, as Michele has highlighted, took the perspective that if you look at other ancestries and other admixtures from a genetic point of view, you're actually going to be able to identify both shared and unique novel genetic associations that may pertain to only one population or another. But what we want to accentuate is that it's also tremendously powerful in terms of helping define those associations that are common across ancestries. So we did a preliminary study that included 10,000 of our African ancestry participants that were meta-analyzed with a largely Euro-Caucasian sample of about 60,000. And just by doing that, we were able to show significantly, a number of significant new findings, as well as fine-tune or fine map findings that had already been significant but were for a broad region. The other thing that I think is important, I mentioned the concept of polygenic risk. Well, when you look at these polygenic risks predictive ability, what we have here is on the left-hand axis, you see what happens when you use polygenic risk that's come from the PGC, which was largely and still is Euro-Caucasian, against another Euro-Caucasian population and has a very high predictive value. The O'Donovan study, again, Caucasian, very high predictive value. There's even some cross-validation with bipolar disorder, which there may well be a shared portion of the genetic liability between these two disorders. But then when you try to bring it to do prediction in an African ancestry sample, and these are from probably 2008, the predictive ability is very, very small. The other thing that this slide shows us is that using the polygenic risk for psychosis is very indicative for both schizophrenia and bipolar disorder. But when you try to use it, when you try to use it for anything else, type 2 diabetes, type 1 diabetes, coronary artery disease, there's no predictive value whatsoever. So polygenic risk is actually something that is relatively specific to a set of disorders. And this was good information to have.
Carlos, before you leave that fascinating slide, you're leaving us hanging here. This is, you had a pretty good size sample in the MGSAA. In other words, you have statistical confidence that this predictive utility is very low. What is that? Tell us something about that. What's your thinking about what that means?
Steve, I think you've hit a good point. I think there are two variables here. One is that it is not easily predicted to go, and I have other slides that with more comparable sample sizes still show this type of phenomena. Part of the signal loss here is scale. So the MGS African ancestry study is considerably smaller than the discovery set from the PGC. So that it's hard to completely define what this is related to, but we clearly see in more comparable studies that you can't just adopt one ancestry as predictive to another.
And just one more question about that before you go on, is this is the same polygenic risk score that was validated in a predominantly Caucasian population?
Exactly.
That is not predictive for African Americans. So you have the potential to say, what would a polygenic risk score look like that was more predictive for the African American score?
Thank you for that slide into our next set.
Awesome.
So this is our own work instead of extrapolating from others' work. And here, what we did is we used polygenic risk that we determined on, and the population sizes are in the ends in the parentheses. And then we sort of started with one ancestry and then saw how well did it predict in others. So for instance, the polygenic risk that we defined from African ancestry was very predictive in African ancestry, pretty poor in Latino, and actually quite poor in European. If we take the Latino ancestry polygenic risk, it's actually pretty poor in African, quite good in Latino, and a little better than we saw with African in the Euro-Caucasian. Part of that is, of course, the Latino population, Latinx population, is admixed more heavily with Euro-Caucasians. The average African American or African ancestry admixed population is at least 80% African ancestry, sub-Saharan African, where in Latino, you might well see a much broader blend, maybe 40-40 with 20 Native American, which is also actually a very important ancestry. Now, when we look at what we did in looking at sort of a meta-analyze, so a trans ancestry polygenic risk, where we combined what we found across ancestries. They were very, and then we looked at how well did it predict it in the PGC schizophrenia sample of about 50,000 at that time. The African ancestry wasn't bad. I mean, 10 to the minus 20, Latino was better, 10 to the minus 52, and 10 to the minus 110 when it was PGC to PGC, basically. When we, and again, I'm sorry, I'm sort of racing ahead. When we truly applied a derived trans ancestry type score, we actually saw significant improvement. So what we found is when we blended the findings from all the three major ancestry groups, they were actually vastly more predictive in any of the ancestry groups, something that others had not done to date. Now, clearly, it's still more predictive of where the dominant population scale of the studies are. But at 10 to the minus 53, you have a very strong signal for African. And interestingly, the meta-analyzed polygenic risk was stronger than even African to African, just because you actually had a much larger scale you were deriving from, and yet retained the information coming from these other ancestries. Again, this is something I'm actually going to skip, but bottom line is it's just showing that trans ancestry does the best. But the minute you start getting out of the broad diagnoses that fit within one of the umbrella, the predictive ability and other things disappears. So there is a degree of specificity to polygenic risk. So what are the conclusions of this type of work? This is not the conclusion to the talk, but to this type of work is one, I think we've clearly demonstrated that there's a shared polygenic basis for schizophrenia across populations. We were able to identify 18 new significant loci with only 4,000 new African ancestry patients, maybe 5. We could clearly improve fine mapping resolution at 22. Michele's telling me I still have 20 minutes, so I don't need to worry, but I wasn't that worried anyway. And we were actually able to identify 4 totally new significant loci that weren't apparent in Euro-Caucasians at all. I think this shows the importance of expanding diverse ancestry genomic studies. And I think the most important aspect of this is an equitable benefit to research and public health. I'm a very strong believer, Michele and I agree on this, that one disparities that underrepresented minorities experience is really the lack of participation in research, without us being able to work with them collaboratively so that we understand the fundamental issues related to the populations we serve. We're really not able to provide the same level of care we are for where most of the studies have been done. All right, this is a purely hypothetical graphic to try to underscore why this is so powerful. So if we take a mutation that happens somewhere in the genome, sorry about that, and you look in Euro-Caucasians, that mutation in a genome-wide association will be found in what's called an LD block, a linkage disequilibrium block. What that means is within the Euro-Caucasian population, that mutation is contained in a stretch of the genome that is inherited consistently across the population. There are recombination breaks at either end, which shift between one person and another, but people who have this particular block will all inherit this block. So once you've identified the block, you can't get to the mutation because you don't know where in the block that mutation is. When we move to sub-Saharan Africa, what you have is vastly more linkage LD blocks. There are many, many more recombinations, and that's based on 2 things. One, the sub-Saharan African genome is much, much older. And a certain amount of recombinations that occur in the genome are totally related to time, the population time that it's had to have these events occur. It turns out there's also some genomic control of recombination, and sub-Saharan African populations contain a particular gene variant that induces recombination much more frequently. That may have some evolutionary benefit, some, again, resilience benefit, but the bottom line is that the blocks are smaller. So what you see here is because it's smaller, you've taken this large block in Eurocaucasians, and now you're defining that mutation is occurring in a smaller area. So you've eliminated sections of that block that no longer are relevant to that mutation. Now, when you're looking at Latinx, you're going to have input from African, input from European, but you're also going to have input from native populations in Latin America and North America. When you have Native American contributions, what's interesting is that population actually has longer LD blocks, less recombination events. But nonetheless, they're different than the other two populations. So this is a hypothetical example where the mutation in Native American populations or in that admixed population is detected at the end of a long LD block. But the overlap between all three is actually highly refining to the actual mutation. So that's the logic behind the approach that we're using in looking at multiple ancestries when we're looking at common variation. Okay, so one of the things that we've been able to bring together in Rutgers is tremendously exciting. So right now, Michele and I, with the GPC population, the 65,000 participants in the GPC, along with Jennifer Michele, who we recruited from Emory, Michele mentioned earlier, who is the world's expert on 3q29, along with Ziping Pang from the Child Health Institute, who's here at Rutgers, and Ron Hart, who's one of the stem cell specialists at the New Brunswick campus that works with differentiation of stem cells into neurons and organoids.
And circuits.
We are in the process of designing a study that is going to look at schizophrenia-related copy number variations. These are very rare mutations. I will highlight the ones we're going to look at. We're going to look at 3q29, 22q, 15q13.3, sorry about that, 16p11.2 proximal duplication, and the Norexin 1 deletion on 2p16.3. So what's fascinating about these mutations is, one, as Michele pointed out with 3q29, they have an actually relatively interesting shared phenotype, which is, I wouldn't have called it as advanced autism. I would say they are highly functioning autistic spectrum children that unfortunately develop schizophrenia very frequently as late adolescents, early young people in their 20s. They have a totally typical presentation of schizophrenia, but what's amazing is if you carry one of these rare mutations, the likelihood, the penetrance is extremely high. You're likely to get schizophrenia at a rate of 70%. The other thing that's really important in this slide, so we're looking at the green spots at this end. When you're looking at what we've been talking about until now, the copy number, the common variation, we're at the right hand of the screen. We're at that tiny odds ratio impact where you have a cumulative impact that probably accounts for 10, maybe ultimately 15 to 20% of inheritance. But for these that we're talking about doing in this next study, we're talking about things with an odds ratio of 170. As you can see in this slide, the ranges are vastly higher than you see with common variants. Now, we are able to do, what is the study that we're proposing to do? our hypothesis is that these highly penetrant rare schizophrenia mutations are likely to have large effects also at the molecular and cellular levels. So what we're proposing to do is we're going to create stem cells for groups of patients and controls that represent each of these 5 rare mutations. Now, as I may not have mentioned earlier, these have a frequency of between 1 in 1,000 patients with schizophrenia to 1 in 500 for the most common. So it's extremely rare. So the idea of constructing a study that that would say we're going to go find these individuals is extremely lacks in credibility. On the other hand, with the 65,000 participants we have whole genome data on, we were able to identify groups in each of these variations and highly genomically controlled controls for them. So what we're proposing to do, as you can see here in this table, is we're proposing to do 15 stem cell lines for each of these deletions. And the majority of these will come from underrepresented minorities.
And the control.
So the controls, as Michele is indicating from the side, are defined as controls by 1, not having any of these deletions or similar deletions that might cause schizophrenia. And 2, we are going to grow these cells in what's called a village on a dish. So we're going to both do the stem cell lines and the then differentiated neurons in an environment that each of the mutations, or each of the individuals with a mutation plus the control are grown together. And we can individually identify the cells derived from each one. So
they come from our 65,000.
What Michele wanted me to make sure I point out is we were able to do this because out of our 65,000 participants, we actually not only have these 75, but we probably have another, maybe an additional 75 with these mutations. Most of the additional 75 are Euro-Caucasians. And we've decided to, again, for representation and generalizability, we want to understand how these mutations, which are shared for the most part across ancestries. You will note that we have no Latino 3Q29 to date. So that may be the one that's slightly different. But the bottom line is we're very interested in how these shared mutations across ancestry play out in each of these genetic backgrounds. And the controls will be able to let us control for that genetic ancestry as well. This is what I was addressing, that each of the plates, let's say we'll end up with 24 plates, will have a different number of patients plus at least one control that's appropriate for the racial and ancestral background. And it's more that we will try to group them so that they're not just same ancestry by, report or by observation, but very tightly close in terms of genetic background, which we're able to do again because we have the genome sequence for the whole genome across these individuals. If not the sequence, we have SNP-based analysis at millions of SNP level. What we're interested in is really looking at testing for convergence. What we're saying is we see them have a shared clinical phenotype. We believe that we might well find a shared cellular and molecular or neural pathway phenotype that will be very illuminating. Knowing that these mutations are totally in different locations on totally different genes, we may well be able to understand on a biologic level what the common pathways biologically are for these findings. I will be happy to take questions on that, but let me just give you a little bit of highlight as we end on some of the other things that we're planning strategically to build. So Michele focused on the center that she runs. We're also looking to create a center for precision behavioral health. We are actually in negotiations to do a cluster hire of an incredibly successful group that does a lot of the same work we do, but in a complementary way that would vastly expand the reach of what we're doing. We're also recruiting an endowed chair as the Rutgers Addiction Research Center Director, and this is in collaboration with the Brain Health Institute. And Michele and Frank Ganassi are working on the creation of a center focused on OCD research and treatment, which would be the first sort of model program to build with RWJ Barnabas. It was just mentioned very briefly that we just announced the groundbreaking on the Brandt Behavioral Health Treatment Center, which will focus on adolescents and young adults, and where there is a $30 million donation to launch that. And we're completing the construction of the Plainfield Addiction Center of Excellence, which is a renovation of an old hospital into a program completely focused on addictions. I think that, as usual, I forget the slides that I have going forward, and already said this. I think that one of the things that I want to end on and allow a little time for questions and answers is that in acknowledging, the most important thing is to acknowledge the GPC study participants and their families who have been with us, incredible collaborators. Michele failed to mention one of the things that she really achieved in the design. We have a nearly 95% consent to follow up these people. For instance, with our Azorian, and we worked in the Portuguese islands at the beginning. We have been following those studies for nearly 30 plus years. And we are now funded to study, unfortunately, the 4th generation of patients with schizophrenia and bipolar disorder in these families, because we started with their great-grandparents, parents, and grandparents, and now they unfortunately are becoming ill as well. We also have just the type of work we do is absolutely team science. And we've been very privileged to help coordinate and fund this work and collaborate with some of the best in the world. So at this point, we'd like to open it up to questions.
That is absolutely awesome. And thanks so much to both of you. I have one question that it was actually stimulated first by Michele's discussion of the differences that may exist in response to medications like antipsychotics. But then I perked up again when you mentioned the Center for Precision Treatment. So a couple of the research questions that some of our work with Mark Olfsen and Scott Stroop and others over the years have been concerned with. You know, at the health services research and service delivery system levels, we're always kind of looking at the kind of treatment that people with severe mental illness, especially schizophrenia, receive and don't receive. And we've done a lot of this work on the appropriate use of antipsychotics and the risks of antipsychotics and potential differential effectiveness. So when you think about and I think some of the methods that you're using, you know, certainly RCT methods could be some of them, but maybe also, you know, when we think about the population health level, some of the ways to put all these amazing methods and data to work sooner rather than later to help people with schizophrenia. I think about some of the questions that we need more information on, for example, thinking about both response to particular antipsychotic and other medications, psychotropic medications, and the risks associated therewith. Right. So for just to name a few of the questions that are floating around out there, could we identify with genetic risk or potential responsiveness scores, those putatively non-treatment responsive people with schizophrenia, right, who might do well on clozapine? Could we identify those who should not be on olanzapine or another very metabolically aggravating
exactly
drug?
And there are already examples of known variants that predict poor response and unfortunately high degrees of side effects. And I said they may well be disproportionately present in one ancestry versus another. But the reality is you could be, in the example we gave, you could be Caucasian and be in the 2% that should not be taking that medicine.
Well,
Right.
So that type of screening is something that is part of the precision
Right.
medicine approach.
So one of the things that's so cool about the stem cell work that we're going to do with Jen is that that's looking at rare variants and how can rare variants help us to understand the common variants are so common it gets kind of mushy. But if we can start at the rare variants and expand up, it can make a huge difference. One of the things that was so exciting for me about the work that Jen and I had done, as I alluded to, was she's the person I had given a whole bunch of samples to early on in her career. And so she came at it from looking at this autism that was developing into schizophrenia and identifying this rare variant. Now we're going to go backwards and look at these rare variants and see what they can tell us about phenotype. So it's really a way of trying to go across.
Right.
So to complement that, do you think there's also potential as we expand the treatment populations to integrate the EMR data with genomic on the larger populations with genomic data, with healthcare claims to look at some of the outcomes that might not be captured in the EMR data?
Well, that's why his bar graph with all the colors was important, because you can see how studying these different races and seeing where they cross over could tell us things.
Yes, but actually, I mean, this is part of the conversation that Nathaniel and we had yesterday, which is the ability to work through the CTSA on large data sets where we can start defining a sort of a discovery set of things that cluster and then test them in the populations on which we have genomic data is extremely powerful. And regardless of what we're studying, it doesn't matter what the clinical phenotype is, that approach should prove to be very valuable.
Right. And if we could get the appropriate consents within large clinical populations to just do their genome and start associating that with some of the outcomes, I think there might be interesting possibilities within the system.
One of the interesting things is because of the way we collect data, we know what our specific aims were, but we have data on diabetes, on cardiac disorder, and so on all these people. And we've done COVID studies already on some of these individuals. And so I think, we're looking at how we might well collaborate with your core and develop interesting programs that the CTSA can help coordinate, and we would be delighted to.
Absolutely. Let me encourage everybody else. I know folks are on mute, but does somebody else want to jump in with a question or a reaction? Nathaniel, what are your thoughts?
I thought it's really exciting. Sorry, I'm trying to turn on my video. We haven't in our own group thought much about genetics except for just a very few candidate genes. And so I'm really thinking a lot about the opportunities of the GWAS and the particular variants of interest and the family structure and so on. And I think I'm particularly interested in, genes related to ward and learning and decision-making, dopamine, which I mentioned in there as well. And I think there's a whole set of questions that one could ask.
Thinking in your space where you like to use neural net and machine learning approaches that maybe extract some additional information from a whole bunch of data than you just get from an additive risk or resilience store. Dalton Conley tells me that I may be overestimating the potential for this, but is there room for machine learning approaches that could take into account not just the additive effects of all these variants, but their interactions?
Yeah, I would think so. I think that's another, actually, as we've discussed in other domains, you and I assume that people who specialize in this area have their own models and so on that they work with and know what they're doing. But I think with these very large data sets, I think there really are opportunities for automatically doing sort of feature selection, trying to discover important interactions and so on. Obviously, the multiple comparisons issues become bigger, but I think that could be dealt with. It's very interesting.
Yeah, and with the GWAS methods that you have, there might be some interactions that would reach those very high thresholds of significance that are kind of allowed in your business. Amasika, I just want to try to get this conversation in our last couple of minutes as broad as possible. Just any quick reactions, how this might apply to the clinical populations, to substance abuse treatment, vulnerability to substance abuse treatment, risks, maybe even potential responsiveness to different treatments?
I mean, I think that for all of those things, one of the kind of more immediate questions that was at the forefront of my mind, maybe it's two, and I might have missed it. One was, are your cohorts closed, or now is it possible for more individuals to enroll? Because I think there's a real interest.
We're still ascertaining.
Right.
Okay. All right. Awesome.
And then the other key feature about that is while our grants are to collect people with African and Latino ancestries, we never turn anyone away. And even now, we have permission from the NIH. If we had more European Caucasians or Chinese or any, the whole idea is to let everyone who wants to participate.
But
are you interested in enriching your cohorts with people who have substance abuse issues?
We don't exclude any. So
yeah, we don't exclude.
We have multiple papers looking at substance abuse, because we're very much interested in looking at real population,
humans.
Not a highly special, controlled and enriched population for a particular thing that becomes non-generalizable and non-representative. So, eliminating substance. Now, if someone's only symptoms were in the process of using a hallucinogenic, obviously that's different. But in general, we have alcohol, all the substances, so we do not exclude for any of those reasons, but we can analyze those.
I can tell you from our claims data analysis, it's hard to find a person diagnosed with opioid use disorder who doesn't have various mental health diagnoses.
And I mean, that's the real life experience. And again, when you're doing population genomics, I think it's extremely important to do this type of population structuring that we're doing to achieve representation.
Just by example.
But I have to add, Michele always says this, that we won't turn down anyone, but we are very focused on underrepresented minorities. And in order to do that, our ascertainment structure leads to very few that aren't going to appropriately fit the population. Whereas originally it was the opposite. We were looking very broad in a population that did not have, for instance, many African ancestries. So we had to do special outreach for that. But
we do have a finesse. We refuse anyone as a control who's had traumatic brain injury. And you can only, but we did even write a paper on this. We don't refuse you if you have schizophrenia or bipolar disorder, as long as those diagnoses preceded your traumatic brain injury. And we wrote a paper where we looked at who had more traumatic brain injury, those with bipolar illness or those with schizophrenia. And guess which group had more? The bipolar group, because they do reckless things. The schizophrenia group tend to get, because of their negative symptoms, they don't do anything. So
at least that's one theory of why. But
anyway, so we do navigate it that way.
Really, really awesome. And I think although you've got plenty of them, plenty of people with adverse social determinants of health and issues of homelessness and severe childhood trauma and so forth. And in our New Jersey populations, you get even more.
Right. We in 2018 we added the ACE, as a screening for all our cases and control. And we already wrote one paper that showed in our OCD sample, those with OCD and those with body dysmorphic disorder plus OCD, the body dysmorphic plus OCD sample had higher ACE scores before age, so more traumatic events before age 18. And that's, yes, no, 10 questions. It's very easy to get an idea of the kinds of traumatic brain injury, not the level,
traumatic events.
Yeah,
But,
yeah,
and then we looked at, guess what? That group also had more PTSD, more traumatic events, more PTSD, you know.
right. Right. Amasiko's work, I think, is going to increasingly sort of
And that's why we've had so much.
lead us towards the role of severe adverse childhood experiences and PTSD in people who are people with opioid use disorder. I'm guessing, and that's
So more collaborations to come. Good.
Yeah, awesome. We should let you go, and thank you.
And thank you for having
Thank
us.
you so
much.
And
Thanks so
please,
much.
please share. You know, it's fine with us that this gets recorded and repeated. Residents might get interested. Whatever.
Oh, it will. It will be quite a bit of viewership that way.